Welcome to Let's Get to the Marks. Grab some paper and a pen and have a go at this tricky A-level biomaths question. Let's go. The two DNA strands of a particular gene contain 168 guanine bases between them. The relationship between the numbers of guanine bases G and adenine bases A and thymine bases T and cytosine bases C in these two strands is shown in the following equation. G equals 4, bracket A plus T minus C. Use this information and your understanding of DNA structure to calculate the maximum number of amino acids coded for by this gene. Show your working. So, how are we going to work this out? We also need to highlight that it said two DNA strands, the sneaky little kids. Right, so what do we know? We know that G equals C, so G is 168, so therefore C is also 168. So I can write this equation. Oh, and I also know that A equals T. So A plus T is the same as saying 2 times X or AT, but I'm just going to write 2 times X. So I'm going to set mine out like this. G, so 168 is equal to 4 times 2x. Why have I put 2x? Because A and T must be equal because they're equal to each other by complementary base pairing. If I have 20A, I have 20T. So I'm just going to call that 2x and then minus 168. So now we just need to solve this. So how are you going to solve this? Well, I'm going to add 168 to each side. So let's add 168. And that's going to give me 336. And that is equal to 4 times 2x. Then I want to divide each side by 4. So I'm going to end up with over here 336 divided by 4 equals 2x. So let me smash that in the calc. 336 divided by 4 equals 84. So 84 equals 2x. So what does that tell me? That tells me that A is equal, and then we need to divide by 2, obviously, 84. And that's going to tell me that A is equal to 42, T is equal to 42. So what is my total number of bases? Well, we know that G is 168, C is 168, A is 42, and T is 42. Let's add it all up. So we've got 336, which is 168 plus 168 plus 42 plus 42. Essentially, we have 420 bases. Aha! Here's the little trick, which when I first did this, I missed this. There are 2 DNA strands, and it's asking us to find out the maximum number of amino acids coded for by the gene. We've got 2 copies of the gene. So what we need to do is divide by 2, and that leaves us with 210 bases. Now, how many bases code for each amino acid? Well, we need 3 bases to code for an amino acid. So I need to do 210 to find the maximum amino acids you can make is 70 aminos because you need 3 bases, i.e. CAG. Well, that's non-coding. So AUG is the start code. So like 3 bases, that doesn't code for an amino acid either, but you get the idea. GGA, there we go. So you need like 3 bases to code for an amino acid. So we want to do 210 divided by 3. The final answer is 70. Now you're going to get 2 marks for coming up with 70 amino acids. If they see anywhere on the mark scheme that you worked out A was 42 or T was 42, you're going to pick up a mark. If you worked out that and it showed any of this working, you're going to pick up a mark. It's like that with biology. It's very easy to get the first mark from the working. It's often very difficult to get to the final mark. You'll have done well if you got to the fact that there were 420 bases. You did pretty well. The hard bit was to realize that there are 2 DNA strands, obviously, of a particular gene. So that means you've got 2 lots of the gene. So you've got to divide by 2 to get 210 and then divide by 3 to get the number of bases. Okay, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.